some of these terms do become a little more easily understood in how they relate to a phylogenetic tree. So we saw that we have a principle called parsimony. Parsimony is not a natural law, but it simply says in terms of a phylogenetic tree that using the available information, you should reconstruct the simplest phylogenetic tree, which has the fewest nodes, in order to account for all characters that you have. It doesn't guarantee it's the right answer, but it is the best approach. And it can be done, of course, by computers, but in small data sets like you might have in an exercise, it can be done by inspection. So we've sort of touched on these ideas of in-groups and out-groups, but the reason that we have to do this is if all you have are data from the group that you're interested in, it's really not possible to determine what's an apomorphy and what's a plesiomorphy. What we usually do then is we have the group we're interested in, and then we try to use as an outgroup something that is closely related to it, but not part of what we think is part of the group. The reason is, let's say, we'll use letters here for some character trait. You can think of it as long tails, short tails, whatever it might be. So let's say this branch has trait A, and these two branches have B. But in reality, we don't know how these three are related to each other because we don't know what's plesiomorphic and what's derived. If a trait shows up in both the outgroup and in-group, then we assume that it has to have been down here. It's a plesiomorphic trait. So a trait that is occurring both in our outgroup and our in-group, we assume is a plesiomorphy because any other possibility would be not be as parsimonious. That's essentially why we have to do that. And when you're actually working through your phylogenetic exercise, that should be clear. Now, for reasons of time, on a test, I'm not going to actually have you reconstruct a phylogeny. You'll have to still be able to interpret and understand all those concepts, but you're not going to be given a data set and say, construct a phylogenetic tree. But in the exercise you are, and we'll work through one of these that shows that using some DNA. DNA methods, of course, have been proven to be extremely useful. It can sometimes get to the point where I think some people who work with DNA forget they're actually dealing with organisms. A lot of the best approaches today are more integrated, where they use both morphology, anatomical characteristics, and DNA characteristics. But DNA characters have been extremely valuable. DNA has certain utility in use. For one thing, if you're looking for homologies, there's nothing more fundamental than the DNA sequences. Problem with DNA sequences, one of the issues with them, is that first they have to be aligned. Alignment means you have to know where to start. And that's not always obvious when you're comparing different groups, because sometimes you might have a situation where there have been, say, deletions. Because in real DNA, you don't actually see a marker that says, two slash lines that say deletion occurred here. You have to infer where deletion occurred by looking for sequences that are similar and lining them up. Now, originally, they would do this by hand, lay out their strips of paper with all of their different sequences and keep on working them until it came out. What would you expect they probably use today for alignment? Yeah, computer programs that look for the most likely alignment with the highest support. Because obviously, if you started here with the first G and started this with the second G, you're never going to get any comparison. So alignment is important. Assuming you have sequences that are correctly aligned, another issue with DNA is that there are only 4 possible character trait states. It can be an A, a C, a T, or a G. There are no other character states possible. Which means that reversal, as we've seen through mutation, can certainly occur. And that can be difficult to detect. You can, if you have you might reconstruct something like this, but of course with only a single G to C transition, but it's entirely possible that you might have had a reversal. G becomes C and C goes back to G. That would be very difficult to detect. And that might actually change the way the relationships actually work. Now, the only way to actually deal with that is simply to use very large data sets. With very large data sets, reversals can be they don't disappear, they just become a smaller portion of what you're looking at, and it becomes more reasonable to do it. Now, in exercises like you'll have for phylogenetics exercise 2, we're using a small data set, but no one would use for a real phylogenetic reconstruction as small a data set as I would give you in an exercise. You would use a very large data set to account for things like reversals, to make sure alignments are correct and things like that. So let's just consider this hypothetical group of 3 species. This is our in group. And can we reconstruct the phylogeny without an out group? And it turns out that we can't. Without an out group to compare, we actually have 3 possible reconstructions here. That is, based on our information, all 3 of these are possible. Now, I realize that you're not working through all of these in your head right now to see why. You'll just have to take my word for it at the moment. But if you did, any one of these 3 combinations could be correct with that data set if you have no outgroup. Part of the reason is you don't know what, in some of these cases, is the ancestral condition, what's the derived or apomorphic condition. We can't tell from this whether the ancestor to this group had a G, which later became a C, or the ancestor had a C, which became a G. This means you really can't work out some of those relationships. We'll see what happens when you have an outgroup. Actually, I'll show you 2 outgroups, but we don't really need both of them. So here we have an outgroup now present. Let's look at this outgroup. Given this circumstance, remember the outgroup is more distantly related. So a character state that occurs in both the outgroup and the in-group is probably going to be basal. So what would we assume was the most parsimonious likelihood of being the ancestral trait? That it had a G in the 3rd position or a C? Yeah, the most parsimonious idea is that it had a G in the ancestral condition. And that allows us to, and we also, incidentally, if we look over at the 3rd group here, what would you infer was ancestral here? Was it the G or the A?
G.
The G. And that allows us to reconstruct. So the one that fits best, most parsimonious reconstruction would be this. Now, remember, a most parsimonious reconstruction doesn't have to mean that it was what really happened. It's possible that 2 things did independently occur, but the most parsimonious one is what we go for if we have nothing else. Yes?
So the outgroup has to stem off from like the very ancestor?
Yeah, we are assuming, again, that if this is what we're interested in, and this is our outgroup, the outgroup and the in-group have a common ancestor. So what we're asking, like there was the outgroup, did it have the common ancestor have, say, a G or a C? And if we see that G's are present in some of them here, and G's are present in some of them here, but C is present only here, The most parsimonious way to do that is for G to be the ancestor. The same thing works with anatomical characters. I'm just showing you here in the case of a DNA example. Now, of course, again, although in a small data set like this, and this is about the size of the data sets I'd give you for the phylogenetic exercise, with some work and some procedure, I haven't given you all the procedure, this can be done by logic and by inspection. If you're dealing with data sets of many species and thousands of sequences, that's what Mr. Computer's for, and then they start bringing in all their bootstrappings and all those other techniques. But this is simply to give you the idea of what's going on. However, there are still some things that can be difficult here. What is in the ancestral condition, T or C? Or can you tell from these data? No, it's equally possible that T was the ancestor and in the outgroup it became C, or C was the ancestor, in the in-group it became C, and C was the ancestor and in the outgroup it became T, because there's either one is equally possible. How would you solve that position? You'd have to have another outgroup. An outgroup, you throw your previous outgroup and make it part of the in-group, and then you'd have to have another outgroup. That's known as an equivocal spot because the information you have doesn't allow you to tell the relationships here. There are two plausible possibilities. That's why it's known as equivocal. But if we have a new outgroup, and so all 5 of these are now our in-group, then what does this tell us must have been the ancestral condition?
T.
T is the most likely ancestral condition. And, oops, wrong way. So this would give us the full reconstruction. Now, I realize, as I said the other day, I've gone through this rapidly. None of this is going to sink in fully until you actually do this sort of work, which is why you'll have a phylogenetic exercise and not a test question on it. But it does actually follow quite logically if you work it through. And the exercises will give you procedures. The first exercise is not DNA-based, but anatomical, with mythological made-up creatures, and the second one is a DNA-based exercise, which I point out the vast majority of students score very well on both. Sometimes get 50 on both because it's fairly straightforward once you get into the procedure. So, as I said, cladistic analysis has had effects on classifications. It can sometimes change our classifications that we have. And using the principles like parsimony, we can actually solve some relationships. One of the things that's of interest is that fish and the difference between lungs and swim bladders. Most bony fish, not sharks, have something called a swim bladder, whereas land vertebrates have lungs. So which is the ancestral condition? Darwin believes, since fish more or less preceded land vertebrates, and modern fish, bony fish, have swim bladders, that swim bladders came first and swim bladders evolved into lungs. It made a certain amount of sense. Simply turns out not to be true. Without going into the details here, if you look at where swim bladders exist and where lungs exist among fish, and there are fish with lungs like lungfish, what you see is that the only way to have the swim bladder as the ancestral condition requires that it would have to have evolved independently 3 times. But if lungs evolved into the gas bladder or swim bladder, that's 2 times. And of course, that's not the only evidence we have. There's other evidence through the fossil record that confirms that. So in fact, it turns out that lungs preceded swim bladders. The first thing to evolve as an outgrowth of the digestive tract was lungs. Probably in fish like modern lungfish and others that live in very stagnant waters and have to come up to the surface to supplement directly with air. It turned out as an exaptation in some fish who lived in areas where their gills were working perfectly fine to be more useful as a flotation item. Sharks and rays actually are have branched off before swim bladders evolved, and they have nothing of the kind. And I already mentioned, of course, the story of the reptiles, but again, just to look at that one more time because it's such a good example of what paraphyletic means. The reason reptiles were separate and birds were considered separate classes was primarily because birds had feathers and no other organism, vertebrates that exist today, had feathers, and it was felt that was a unique defining characteristic. Basically, it was assumed it was some sort of synapomorphy that separated them from all other vertebrates. Reptiles were actually not defined, however. There isn't really any synapomorphy of reptiles that doesn't include birds. They were defined by very general traits. There were actually very ancient plesiomorphies, things like that they had internal fertilization instead of doing what frogs do, spraying their eggs over, their sperm over eggs in the water, or the fact that they had scales, or that they had a four-chambered heart, which of course some do, or that they had eggs that had amniotic fluid. None of these are really what we would call synapomorphies. They're all plesiomorphic characters. And ultimately, it became more and more clear that the only way to resolve that was to recognize that birds are nested within what was traditionally called reptiles. And specifically, birds are within the clade that includes dinosaurs, the saurians, and more specifically certain groups like Tyrannosaurus and so forth. They're more closely related to a tyrannosaur than they might be to, say, the large brachiosaurs and other things like that, or some of these dinosaurs over here. Molecular clocks, one of those things I tend to delete some of my discussion of molecular clocks when we lose days to storms and so forth. So let me just say what I'm referring to. We won't go into the nuts and bolts of how people come up with molecular clocks. The idea of a molecular clock is, can you not simply reconstruct an evolutionary history, but can you actually put a timeline on it, like when groups split from each other? It's one thing to say that reconstruct your phylogeny and say that these two groups diverged. How do you know when that happened? Well, one way to determine that, and always the best way, is if you have fossils, because fossils are real physical things and you can put a date on them. But we also like to do that when we don't have fossils. And the molecular clock idea, beginning back in the 1960s, was based on an idea that if you have a predictable rate of mutation, if you have groups that diverged, at this point, of course, it's just one group. The more they diverge from each other, mutations will make them less and less similar. Does that make sense? If you know the rate of mutation, how many mutations occur per 1,000 years or whatever it is, you can then calibrate and say they must have separated so many years ago. All of that is perfectly fine as long as you meet all the assumptions, like you know the rate of mutation. It's usually based on things that are more neutral, not under strong selection, where the mutation rate is random and you can look and estimate it. But the problem, it does give useful dates, but often with quite a bit of spread depending on the assumptions in the gene used. This molecular clocks work best when you have certain points where you can look at an actual fossil event and say, or geological event to calibrate them. So later in the semester, when I show you some of the human evolutionary steps, particularly when our ancestors diverged from things like chimpanzees and gorillas, since there's no fossil evidence of that, you'll notice there'll be huge, big spreads in the dates from molecular clocks because it depends on assumptions. Good things to calibrate, like to see where New World monkeys separated from Old World monkeys, there's a molecular clock date. But since we know when Africa separated from South America, that allows us to come up with a calibration and see, is the molecular date reasonable? But if you don't have that, as we don't try to decide when humans separated from chimpanzees, you're going to get a wide range. So molecular clocks are useful, but they do have to meet some assumptions, and they're only as good as the assumptions that you have about your DNA. All right, well, our last topic for this test unit is population genetics. And I have essentially taken what are 2 chapters in your text and treat them as one. Now, I always recommend, and I still do, of course, and it's important here that you look at your textbook, but I will point out that the 2 chapters in this textbook have a lot of mathematics that won't be in this course. I will cover the Hardy-Weinberg mathematics, but that's only like at the beginning of these chapters. For every new concept they introduce, they'll start throwing out little boxes with all sorts of equations. And if I wanted to cover that, and I really don't, but if I did, I would have to take probably several weeks on it. And there's an old saying that goes back to Hawking's that every time you do something, for every equation you put in, you assume you're losing half of the remaining people. So we'll stick with only one equation, so I'll only lose half of you. No, it won't be that bad. So the rest of it we will be covering pretty thoroughly, but not in a mathematical sense. So this is about population genetics. So we are dealing with populations. In Biology 101, you probably, and maybe a little bit 102, you were primarily looking at the genetics of individuals, maybe a little bit of introduction to population genetics definitions in 102. Don't really know to what extent genetics class does population genetics. The focus in population genetics, as the name implies, is not on the genetics of the individual, but the population or the species. Remember, a population is a unit of organization in which a population consists of individuals of the same species. There is a higher level of organization. It's an ecological level, which is not one we need to use in this course, but some of you are in the ecology course or have been. It's the community. There are levels above that. We don't need to talk about those. In population genetics, one does come across the word gene pool. It's not a word we'll be using a lot here, but a gene pool is a population concept because it is all of the genetic information in the population. So you take all the genetic information in a particular population, that's the gene pool. And what we need to focus on here in a population is, if you remember the differences as you should, what an allele is. Remember, mutations can form new alleles. In a population, you can have an allele frequency. If you went back to your old basic genetics, capital A and capital little a are 2 different alleles, and remember, we weren't doing any Punnett squares in this class, but you know what those are. You could come up with various combinations of possible offspring. Each of those would have a frequency in a population. If we envision some gene like that has 2 alleles, like the little finger straightness gene in this room, if we had 25 people, there would be 50 copies of that gene, and there could be anywhere from 13 and 12, 13 capital A, 12 little a, 49 of one, 1 of the other, hypothetically 50 of one and 0 of the other. They can have all sorts of different possibilities. And of course, you may have more than 2 alleles in a population. There may be 3 or 5 or 1,000. So each allele will have a frequency. And what must they add up to? Yeah, they have to add up to 1.0. So it could be 0.6 of these and 0.4 of these, and they would add up to 1.0. Those are frequencies, and this is what we use. Of course, percentages would mean the same, although traditionally they're done in terms of frequencies. So the frequency of all of the alleles in the population for a particular gene have to add up to 1.0. There are also genotype frequencies. These are 3 genotypes, and one could, in a population, determine what proportion of the population is represented by each genotype. And again, they must add up to 1.0. Obviously, there's a relationship between allele frequencies and genotype frequencies because each of the alleles has a frequency. And in the end, you get genotype frequencies. So what is our definition of evolution in these population genetics terms? As opposed to descent with modification, we say that evolution occurs when allele frequencies change. And we do have to specify from one generation to the next. By definition, evolution is a change in a population over generations of allele frequencies. So in this genetics terms, if the allele frequency of capital A goes from 0.554 to 0.556, that's evolution. Is it a massive change? No, but it is an evolutionary change. It's not inconsistent with descent with modification because a change in allele frequencies is a modification of the allele frequencies. It's simply looking at it in genetics terms. So any change in allele frequencies is an evolutionary change. This was worked out fairly early in the late 1800s and early 20th century by people trying to work out population genetics. But their problem was that they weren't mathematicians. In fact, many early biologists didn't do any mathematics. So they tried to, and your textbook describes this a little bit, they were trying to figure out how evolution worked on a genetic level. They just couldn't get it done because they didn't understand mathematics. So some of these geneticists trying to bring genetics into evolution, finally, I guess out of desperation, went to actual mathematicians. And the two that they went to, this was independent of each other, but we unite them now, were Hardy and Weinberg, but in English we usually call them Weinberg. This is just to remind you of basic, again, what the life cycle is. So here's Hardy and Weinberg, or Hardy-Weinberg. So Weinberg was German. I don't know too much about him, how he arrived at the answers that we're seeing here. But he's looking very much like a German professor of his era. That's Hardy, and I'm not entirely sure why he wanted to be photographed though. Remember, this was before people were running around snapping photos on little cameras. This was probably involved bringing in a professional photographer from the university and having him pose, and I guess this is why he wanted to pose. He wanted to pose that way. It's kind of an interesting photo. Anyway, he was a mathematician, and they basically came in and said, we have this problem, we can't solve it. He solved it in like half an hour, and they never even thought it was important. It's primarily what he's remembered for. It was just some trivial, obvious thing, because it was basically algebra. But of course, and so it was a silly little problem to him, but it has become known as the Hardy-Weinberg dynamics. Weinberg also did the same thing. I don't know the story there. Hardy, I think it was English, but I'm not sure on that. All right, so what were they trying to do? Well, the idea was they were looking for what would cause evolution to occur. Okay, we've seen natural selection, but is that the only thing? What could cause evolution to occur? Which means what would cause allele frequencies to change? The way mathematicians approach things like that is through the null model. If you want to know what will cause allele frequencies to change, the way to approach it is to ask what would have to be true if there is no evolution. If you have an allele frequency, what would have to be true about that population for there to be no change in allele frequencies? That's a null model. What would be true if there's no evolution? And from that, you can work out the different things that cause evolutionary change, that change allele frequencies. They were interested in doing something else beyond that. It has to do with the ability to use allele frequencies to predict genotypes. What they established, and I'll use Hardy's as the source because I know his story a little better, is basically been called the Hardy-Weinberg equilibrium, which means a Hardy-Weinberg equilibrium means that there's no evolutionary change because allele frequencies are not changing. So if a population is in Hardy-Weinberg equilibrium, two things are true. It is not evolving because allele frequencies are not changing. And the allele frequencies can predict the genotype frequencies. Those are two different aspects of the equilibrium. No evolution is occurring, and allele frequencies predict genotype frequencies. And since the population is not evolving, even if something changes the allele frequencies, it will go back to the equilibrium in one generation. In order to do this, their null model ended up with 5 assumptions. I'll tell you why I put an asterisk by one of these in a bit, but let's look at these. The first 4 are the assumptions that would have to be true about the population for there to be no change in allele frequencies. One of them is that there is no natural selection. Natural selection is having no effect on the population. The 1,4 allele is not any more likely to survive and reproduce than any other allele. No advantage for one allele or disadvantage. We can't have mutations because a mutation adds an allele and automatically changes an allele frequency. It wouldn't change it much, but it does change allele frequencies. So we can't have mutations forming new alleles. The population has to be closed. Technically, that means there's no one entering from the outside bringing alleles in, and nobody's leaving the population. So it has to be isolated, not connected to any other population with individuals moving in and out of the population. And mathematically, it has to be infinite in size. Now, that's a mathematical requirement. Can a population be truly infinite in size? No. But it can be big enough so that it's effectively infinite. All 4 of these would have to be true. Mathematically, all 4 have to be met for there to be no change in allele frequencies as a result of evolution. The 5th, panmixia is a technical term. It means, for sexual species, random mating. Literally means all mixed up together. Sounds like some sort of giant orgy or whatever. It means no one's making any choices in mating. Who one male mates with is totally random, and who the females mate with, or they're hermaphrodites. Whoever they mate with is random. You're just pulling them out randomly and sticking them together. So no mate choice. That's not relevant to allele frequencies directly. It is not, no, it's not an asterisk because that's not a force of evolution. Panmixia is required for allele frequencies to predict genotype frequencies. So whether mating is random or not is not directly a force of evolution because it doesn't involve changing allele frequencies. It is necessary, however, for another thing they wanted, which is for an allele frequency to predict a genotype frequency. Okay, we'll take a couple minutes break and then we'll dive into looking at the actual mathematics of Hardy Weinberg. That was an interesting